Alright everybody, so what I'm going to be taking you through are the steps for transcription and translation that the cell goes through in order to produce a protein. So first off, I'm going to show you all the players that we'll be working with today. So this right here, these two strings represent the sugar phosphate backbone, and then all of these are the nucleotides all the way down, and you can tell they're the nucleotides because you got the G's with the C's, A's with the T's, T's with the A's, all the way down. Then this strand over here, this is our mRNA, and you can tell because there's U's instead of T's that will match up with the A's, as you'll see in this demonstration. This guy right here is our RNA polymerase, mRNA polymerase. And the way that I always remember what polymerase does is it unzips the DNA strand so that mRNA can be formed, and so I turned it into a zipper. This black shoestring right here represents the nuclear membrane, so everything that takes place this side of the string is in the nucleus. Everything outside is in the cytoplasm. So that takes care of everything in the nucleus. If we go outside in the cytoplasm, this right here, it's our ribosome, but just for purposes of this demonstration, we're going to separate it. Then up here we have our tRNA molecules, and attached to them is the amino acids. So we'll get started. The first step here is transcription. So what happens is the mRNA polymerase comes here, just slides down the DNA strand, unzipping it. So as it unzips it, as it goes down, the mRNA is formed. So and this comes here, and it pairs up with the corresponding nucleotides, if they would stay on the right side. So then what you get is the C's with the G's, A's with the T's, and then the U's with the A's. And so this is when our mRNA is formed. And then as it goes down, it keeps on going, and it goes outside of the nucleus. So just like that. And then once it is outside the nucleus, this DNA strand just comes right back together. So that is what happens during transcription. So that's the first step. And then once outside of the cell, or the nucleus, in the cytoplasm, this mRNA strand travels through and finds a ribosome. Once it finds a ribosome, this is when translation starts. So the mRNA travels through the ribosome, and the ribosome reads 3 nucleotides at a time. So first it's the CAU, and then it sends a signal out to the cell, or out to the tRNA molecules, and it finds the corresponding amino acid. And then it travels through and hooks up right there. And once they have the right amino acid, it just keeps on going through to the next 3. And we have the GAU, same thing happens. The tRNA finds the amino acid that goes with that 3 series right there. So it links up. These amino acids stay together. They stay linked up through the whole process. And then the ribosome reads the next 3. Same thing. tRNA brings that amino acid right there, and it links up with the other 2 and just keeps on going down. These amino acids stay together like this. And then finally the ribosome gets to the last 3 nucleotides. Same process. tRNA grabs the amino acid, comes here, links it up. And now what we have is this strand of amino acids. The SER, ARG, ASP, and the HIS. So those are all linked up, and those amino acids that are linked create this protein. Like, that is what the protein is. And so that is how the 2 steps with the transcription and translation leads to the linkage of the amino acid, which, when it's all said and done, is the protein that it set out to create. Thank you.